Okay. Yeah, so I'll record this and obviously you guys can watch it back if you want to. I don't know that it will be that exciting you want to do it twice, but hopefully if Zach has an opportunity to, he can jump on and catch up as well. All right, so we'll work our way through. So the main focus overall is to understand the primary role of our immune system. What does it actually do, and how that relates to antigens. And so we'll unpack some of the key terms around that. Obviously, we've touched on pathogens, but we'll need to talk about what we mean by antigens and both self and non-self. So we'll learn about self versus non-self and the different types of pathogens. And MHC receptors in particular is something that we'll talk about here. MHC receptors are really important in terms of identifying self-antigens. So all the cells of your body express antigens, and that is how your body recognizes you as you and hopefully doesn't attack you. And then you've got non-self antigens, which are typically what we associate those with pathogens. And we should be able to describe the difference between cellular and non-cellular pathogens, which, I mean, the name kind of tells you most of what you need to know, but you'll need to be able to identify which ones are which. Go to the next slide. Okay. For your study design, I don't think I highlighted this one, but we're looking at this bottom dot point here. Well, parts of it anyway, not so much the initiation of the immune response. We'll look at that as we work our way through the other chapters, and certainly antigen presentation. That's more in, that'll be in chapter 7D, I suspect, which will be the acquired immunity. But this first bit here, the distinction between self-antigens and non-self-antigens and cellular and non-cellular pathogens and allergens is sort of the key thing that we're going to look at in this chapter.
Nice of you to rock up, Jet.
I think we can teach him some biology, it's fine.
You want to learn some biology, Jet?
Yeah. So,
You want to learn some biology?
Say hello, don't be rude.
Hello.
hello. They're going to tell you all about antigens after this. They'll know everything. I hope. We'll find out when we do some of the questions in a bit. Yeah, so you want to know by the end of this chapter, you should know the difference between self and non-self and how we can actually, how your immune system can identify the two, and the nature of cellular, non-cellular pathogens. How does an allergen differ, do you think?
What are you allergic to?
Yeah, and you'll learn about some in particular. So either of you have hay fever or get hay fever?
Yes.
Yeah, so no, you're lucky. Yeah, I don't get hay fever either. But an allergen, so in that case, it's pollen, so pollen particles trigger an immune response, which is the allergic response. It's basically an allergic response is an overreaction of the immune system to something that's a minimal threat. If you think about the symptoms you get from, we'll talk about this more later, but if you think about the symptoms you get with hay fever, you come in contact with pollen, it triggers the immune system. What it's trying to do is protect you. So runny eyes and nose, it's trying to flush the pollen out. It's trying to stop anything else from getting in. It's a little bit like, shutting the barn door after the horse has bolted because the pollen's already inside you. But that's what it's trying to do. It's putting up the defenses to try and stop you from getting any sicker. We'll go through that when we, it's the same actually when you get, like, say for me with COVID at the moment, like coughing is trying to expel stuff, like get the pathogen out of me. Blocking my nose is about trying to stop other stuff getting in. Again, it's a bit pointless because it's already in me. It would have been nice if you did that before I got it, but that's kind of what it's trying to do. That's your immune response is literally trying to eliminate whatever is in you and prevent anything else from gaining access to make you sicker. That's really what the whole goal is. Unfortunately, creating a fever, the fever is designed to cook whatever the pathogen is and try and kill it, make it too hot for the pathogen. Obviously, sometimes that makes you pass out and break your tooth. So it's not always good. Yeah. I prefer it when you flip me off because at least that's supposedly a mark of respect. I don't know about the L on the forehead. Yeah, much better, thanks. Luckily I've recorded that, so you can expect to be suspended now, so that's fine. Okay, so we'll move on. I won't play this video because it's stupid. I would have if we were in class. You would have had to sit through it. We could use it as a brain break or something to break up remote learning. There's some good science in it, but it's you'll see it's kids' health. I'll play the first little bit. Hopefully the audio comes through. I assume it will.
Can't hear.
Can't hear? You're not missing anything. So we're going to move past that anyway. There'd be a button here somewhere that lets me do that. I have no idea where it is. There's buttons everywhere and I don't care enough. I could hear it. It was unpleasant. I might have to, there's a video I probably have to share with you guys later because both of you might appreciate it, but I'll have to work out the sound.
Is it a song?
No. Oh, there is a song involved, but I know I want to build up suspense. But I'll need to email. Hang on, I need to email it to myself because I'm on my phone. So bear with me. Well, I think hopefully I can email it to myself. I'm about to find out. Yep. All right, so I'll do that in the background. Okay, let's learn about stuff. All right, so we've already talked about a pathogen. So a pathogen is really anything that can stimulate an immune response and cause disease. All right, so you could actually, we do typically separate allergens that we mentioned and pathogens, but in a sense, an allergen is still a pathogen. You can't even really say a pathogen is a living thing necessarily because viruses aren't living and prions aren't living, but they absolutely can stimulate and cause disease. So it's really anything that is capable of causing disease. What do we mean by antigen?
What picks it up?
Be a little bit more specific though.
That's all I've got.
That's okay. That's the purpose of this chapter. So we'll spend some time on it. What do you think, Flick? What do you think an antigen is? Like you know, rat tests. It's rapid antigen tests. What are we picking up?
The bugs.
Yeah. Basically, yeah. So an antigen is a structure on the surface of a pathogen that is detected by the immune response. It's capable of triggering an immune response. So typically they're proteins, but they can be a range of other things. We'll talk about that, unpack that as we go. But basically they're markers on the surface of the cell that indicate it as not belonging and can trigger an immune response. So they can either trigger an immune response or, in the case of our own antigens, signal that it's actually okay and that's supposed to be here. So antigens are basically markers. And then you've got the two underneath that, self-antigen and non-self-antigen. So self-antigen is the marker on our own cells that tells the immune system, this belongs to me, therefore don't attack it. And obviously sometimes that goes wrong and you get autoimmune diseases. So for example, with me, I've got rheumatoid arthritis and my immune system attacks my joints. That's something's gone wrong there because I shouldn't attack my own joints because the cells of my joints actually have self-antigens on them, which should be the trigger to my immune system to leave them alone. And we'll look at that in a second. A non-self-antigen is obviously a foreign antigen. We typically associate that, and for the most part, we talk about that with respect to pathogens. You know, a bacteria invades causing infection. It obviously has antigens that are not self, and so then the immune system targets it. But other things like hopefully none of you ever need this, but if you needed a transplant at some point, this like let's say it's a heart transplant, so you've received a new heart, the antigens that are on the surface of that heart are not going to match your antigens 100%. They'll try and get as close as they can, but they almost certainly will not match 100%. So your body will recognize non-self antigens on that heart tissue and will try to target it. That's why if you've ever known anyone, or even if you've heard about people who've had transplants, they go on anti-rejection medication. Basically, that medication is trying to suppress the immune system because the immune system will try to kill those non-self antigens that are on that heart tissue or whatever the organ is. So non-self antigens are anything that doesn't belong to you. Oh, I know it's not that exciting page, but crikey. And an immunogen is really anything that can trigger an immune response.
You said crikey.
I did. Yep.
You're taking up my lingo.
Yeah, I'm going to try and work. Yeah, well, that's like last year when I was calling people pelican, which is clearly, you know, so I've grown up past that. I've moved past pelican, but, you know, crikey, I'll have to bring out a little bit of cheeky and maybe some naughty as we go as well, but we'll see how we see if I can work that into the conversation. All right, so what's the purpose of our immune system? It's a scanning system. So your immune system is always on alert. It's always checking for invasion. But it needs to leave your own cells alone. If it starts targeting your own cells, as I've already said, that's autoimmunity. Auto meaning self. So autoimmunity means that you're attacking yourself. So to do this, it must recognize what self is. Like, so how does the immune system actually know that the skin cells you've got, that the heart cells that you've got, like all those cells are actually yours and not to attack them? And a lot of that's done very early. Like immune system is basically presented with the antigens that belong to you as while you're a baby. And so as your immune system develops, it actually will be tested and it'll be shown these various antigens from around the body and go, yep, that's normal. That's mine. I shouldn't attack that. Yes, Paige.
So if someone's pregnant, do they have is a baby? What's that? How does it not attack that?
It actually can. So generally speaking, like the antibodies, which will go when we look at chapter 7D, so that's the adaptive immune response, and that's when you start making antibodies which attack pathogens and or foreign tissue as well, like heart transplants and stuff like that. So we'll look at that in more detail then. But generally speaking, the antibodies don't cross the placenta, so they don't target the baby. But have you ever heard of hemolytic disease of the newborn? Oh, so Flick has, but you haven't, Paige. So hemolytic disease of the newborn is literally, do you guys know your blood group? Let's start from that.
No.
No. So I think you might have told me before, Flick, you're negative? Yeah. Yeah. The AB, O bit doesn't matter. It's positive and negative that matters. That actually puts you at potential risk of, well, not you so much, but if you were to have a baby down the track, potential risk, but we're well across that and we'll do all kinds of stuff, so you won't be. But
You've actually told me that before, so.
Yeah. I just don't want you to go, oh, I can never have a baby. No, that's not the message to take home from here. But when your blood group is negative, if your partner is Rh positive, so they're whatever positive, if the baby that you produce has positive as well, what typically happens is the baby cells can cross the placenta. So some of the red blood cells will cross the placenta and enter the mother's circulation. They're foreign, right? So they've got this positive antigen on them. The mother doesn't have that. So the immune system on the first pregnancy, the immune system just recognizes them and goes, that's not meant to be here, mops them up, gets rid of them, and that's it. But the immune system, and you'll see this when we learn about the immune system in more detail, it keeps a memory. So it remembers that it's seen that before. So typically the first pregnancy, other than the mother being immunized against the positive antigen, nothing happens. The baby's fine. On the second pregnancy, if the baby's also positive, this time the antibodies can start to cross the placenta. So the mother will recognize the baby's red blood cells again, and instead of developing a response, she'll give an overwhelming immune response. So there'll be a massive bump in these antibodies and they can cross the placenta. They're small enough to get across, and they start targeting the baby's red blood cells in the baby's circulation. So they start destroying the baby's red blood cells. If you leave it unchecked, yes, it could. But generally what we do, if we know that a mother is, and we do blood groups on every pregnant, well, as long as they show up for their antenatal tests, we do a blood group on every pregnant woman, so that if we know that she's Rh negative, then we give her, we actually give her antibodies, artificial antibodies. We give her an injection with those. That mops up any of the baby's red cells, so she doesn't become immunized, and therefore she won't attack the baby. But yeah, if you don't do proper antenatal checks and stuff like that, and unfortunately some mothers don't show up for those checks and stuff, that's when you can run into problems. Usually it's not bad enough it'll kill the baby, but the baby will come out very jaundiced and yellow because it's busted open all the red blood cells, and the bilirubin, which is released from the cells, causes the skin to go yellow. And they stick them under, well, they might be anemic, so they might need a blood transfusion. And they stick them under UV lights, which helps break down the bilirubin and sort of get everything out of the system. Usually that's what happens. But we'll learn about that more later, and I'm going to cough. The cough is the suckiest bit. Everything else is fine. I didn't have a cough. Didn't you? Oh, I do. It sucks. So.
I have a question before we move on.
Yes. Yeah.
Are you wearing your socks?
Yeah.
See?
Yeah.
Paige isn't wearing her socks today.
I am. You just can't see them.
Well, I'm afraid you're going to have to lift your foot up in front of the camera, which will be very elegant and. See, it's entirely possible. It's Tuesday. You know the rules.
I forgot.
Anyway, that's all right. Panda socks on Friday.
Yeah.
Even if I don't care if they come halfway up your calf, that's irrelevant. It doesn't matter. Oh, the heel comes halfway up your calf. All right, we'll keep going. So antigens, what is the immune system checking for? It is looking for antigens. So again, they're the cell surface markers. So they're usually proteins, but can be sugars, even DNA with respect to viruses in particular, that are on the surface of pathogens. So they may be attached to the surface, and they can also be free floating. And we have self-antigens on the surface of all of our body cells. And so non-self antigens are important because that's what triggers the immune system to recognize that there's something invading. Won't change. All right, so in order for the immune system to not target you, we have these markers called MHC markers. Right, so there's MHC class 1 and class 2. You don't have to worry. It does stand for major histocompatibility complex, but you don't need to know that. It's just MHC. So MHC class 1 and MHC class 2. The main thing you need to know and remember about these, or every nucleated cell in your body. So the only cells that don't have nuclei, or do you guys know? What are the only cells in your body that don't have a nucleus?
Red blood cells.
Yeah, mature red blood cells don't have nuclei. So every other cell of your body has nuclei. Every single one of those has these MHC class 1 markers on them. So every single one of them. MHC class 2 are only present on immune cells. Right, so they also have MHC class 1, so they have both. Immune cells have both, but MHC class 2 are only on immune cells. Becomes important in terms of how that works later, like how they will identify foreign cells, but also flag themselves as being part of the immune system to help minimize destruction. But we'll talk about that as we work our way through. So MHC class 1, every cell. MHC class 2, only on immune cells. And at the moment, that's probably the main thing you need to know. Excuse me. And that's just a diagram from your textbook that shows that. All right, so every nucleated cell has these class 1s. Your specialized immune cells have both. That's pretty much it. At the moment, all you need to know is that the self markers are MHC markers. They basically your immune system. It's almost like a greeting, like a handshake. If I go back to that, the immune cells will often come in contact with these markers, recognize the marker like a handshake. And so it sort of shakes hands with the marker and goes, righto, yep, that's me, and moves on its way and doesn't do anything. These become important. You'll see this with the innate immune system. Oh God, I'm going to cough again. Okay, it's going really well. Apparently talking is hard. That's okay. Okay, so if your cells are healthy, if your cells are normal, so non-cancerous and non-infected, right? Those are probably the two main things that you're thinking about there. These MHC markers are intact. So when the immune system shakes hands with it, it goes, yep, recognize you, beautiful, move on my way. What happens with some cancers and what happens with a viral infection is these MHC markers get damaged or hidden. And so that means that the immune system tries to shake hands with it, goes, oh, hang on, I don't know what's going on here, but you're not normal, and it will kill it. All right, so the MHC markers are important in terms of recognizing self, but if those MHC markers are damaged, that's when you're going to kill that cell. So you probably know viruses, for example, they work by infecting, like they get inside cells. They're an intracellular parasite. They can't just multiply on their own. They have to infect a cell and live inside a cell. In order for the body to eliminate them, they have to recognize that cell has been infected. And one of the ways they do that is this marker gets damaged and they'll go, hang on, something's not right here. They kill the cell, taking the virus with it. We'll learn about that more as we work our way through that. Yeah, so that's probably all you need to know, that you've got these self-antigens and they're called MHC markers. I've already touched on this, but the three of us will all have different MHC markers. So the ones that are on my cells will be different to the ones on your cells. That's why it's not like we can just say, oh, I need a kidney, you've got one to spare, can I have it? Because we probably won't be compatible because of these MHC markers. All right, so your immune system recognizes yours and says, yep, that's your kidney, leave it alone. But if you pick it up and stuck it in me, it's not going to work like that. So that's important too. Okay, so non-self antigens, they come from outside the body. They're recognized as foreign. So invaders like bacteria, viruses, and fungi, we're going to look at those in just a second. And they stimulate an immune response. So the main source that we consider in BCE biology are pathogens. Yeah, we talk about allergens and other stuff, but it's really the pathogens we're talking about, and they're agents that can cause disease. They're classified as we've already talked about, cellular or non-cellular. So cellular pathogens consist of one or more cells, so they can be unicellular like bacteria, or multicellular like some protozoa, worms, and stuff like that. Yeah, that's all you need to know there. Zach will be devastated because that's Neisseria meningitidis, which is meningococcus. But anyway, what are you going to do? He can just play this video back over and over.
Isn't
and just
that
play,
what you have?
What, meningococcus?
Yeah.
I'd be a lot sicker if I had that. I'd be in hospital. You don't really need to know all the specific details though of like all this stuff, this detail that's from the table in your textbook. So it's on page 357. But you need to understand that they are cellular. So be able to identify that bacteria, fungi, worms, protozoa, they're made out of cells. And that's probably the main thing you need to know. Yeah, you're unlikely to need to know much more detail than that. Non-cellular pathogens are not made of cells, as the name suggests. So viruses are basically genetic material covered in a coat. So DNA or RNA. So some viruses have DNA, some viruses have RNA. And they've just got an envelope around the outside of them or a coat around the outside of them, sometimes both. Often we'll pick up the protein. So for example, with COVID or coronavirus, you would have heard about the spike protein all the time. The spike protein, you haven't heard about the spike protein?
Nope.
You need to watch more news. So the spike protein is literally a spike that's, if you've seen a picture of coronavirus, it's called coronavirus because it looks like a crown. That's actually the reason behind the name. And they've got these spikes all around the outside of them. The spikes actually help them stick to cells and then inject their RNA into our cells. That's how they work. The vaccines that you get, they're designed to target that spike protein. Okay, because if you target the spike protein, it prevents the virus from actually sticking to your cells, and therefore it can't infect you, it can't invade. That's the whole concept behind it. Apparently it doesn't always work. I've had 3 doses and here I sit. But, you know, I'd probably be a lot sicker if I didn't have the vaccine, is the assumption.
Do you know where you got it from?
Absolutely no idea. It's either school or gym. It'll be either school or gym, but that's the only places I've been. So.
Can we see a dog?
She's outside at the moment.
Can we please see it on a brain break?
Oh, we'll see how we go. Let's see how we go. I'll see what she's up to. Yes. So anyway, viruses. Let's talk about those though. So viruses are, because we're almost at the end of this chapter already. So viruses are literally genetic material wrapped in protein. That's it, right? So there's no cell, there's no nucleus, there's no organelles, there's no nothing. They're just genetic material. They are not alive either. The way they replicate is by inserting their genetic material into your genetic material. So they basically genetically modify you. And then your cell reads their DNA. Because remember, DNA is a universal code. Your ribosomes don't look at it and go, oh, I better not read that. That's from a virus. No, they're not smart enough to do that. Just reads the code, makes the protein, and that reassembles the virus, and that's how they reproduce.
How do the viruses get made up if they're not alive?
Yeah, and how do they know to like stick to the other bits?
How do they know to stick? It's all based on receptors, right? So the spike proteins in the virus have evolved so that they, using COVID as an example, the spike proteins have evolved so that they match receptors on the surface of cells in our respiratory system. So they basically, it's not quite exactly as tight a fit, but it's like a lock and key. So it's just really through trial and error. Like and even if you look at COVID, you know, the later strains were much more infectious and potentially more damaging than the early ones because they changed themselves to be better evolved, if you like.
How do they do that when they're not alive?
Just through genetic. It's just through error in copying, right? So if you think you will have literally millions of viral particles in you when you get infected, and you produce millions more. Every time you do that, you are reading the DNA code of the virus. Every time you do that, there's a chance for a mutation to occur. Now, some of those mutations will be useless. It'll make the virus less transmissible, less damaging, less able to invade. Well, that strain, that'll die out. That would have absolutely happened with COVID. There'd be strains of it that evolved as a result of mutation that were no good, and they just didn't get passed on. But there'll be other strains as they evolve, they develop better ability to be transmitted and to invade. And so it's just trial and error, which pretty much all of evolution is. like all of evolution, we haven't done evolution yet. It's probably, we're talking about it back to front a little bit because we do evolution after we do immunity. But evolution is literally DNA mutations is what drives it. Mutation causes variation. The strongest variations survive. The ones that are weaker die out. They accumulate, for us, it takes millions of years and they accumulate over millions of years and we change. But viruses replicate, within days. Well, within hours, actually. So every few hours, you have a chance to make a brand new virus. The other advantage is that with COVID or influenza does it too, because they have other hosts, not just humans, they come in contact with other viruses and they swap genetic material and all kinds of stuff. Which
A
is
little bit naughty.
Yes, definitely naughty. Well done working that in. So we've just, I think I've got my little bingo card here and we've managed to hit crikey and naughty. We just have to do cheeky now and we're set. So that's viruses. We'll talk more about those and how they work and how we kill them, how our immune system kills them. But yeah, they're not alive. They're not sentient, so they can't, like, how do they know to invade? They don't. They just literally are passed from one person to another. Obviously, like anything, its goal is to survive. They don't want to kill the host. If they kill the host, they kill themselves. So, if you die...
How do they know not to do that when they're not real?
Well, they don't. They don't. And the thing that kills you is not the virus. Like, say, with COVID, the virus doesn't kill you really. It's your immune response kills you. We'll look at how that happens when we look at the specifics of the way immunity works. But literally what was happening with people who were dying, their lungs would fill up with fluid. So inflammatory fluid, basically trying to kill the virus. It gives you an over-the-top response and you, for want of a better term, you suffocate on the inside because your lungs are full of fluid, you can't breathe. But it's not the virus. The virus triggered the immune response. So the virus is still responsible, but actually it's not the virus that caused that. That was that's the immune response that caused it. Which is no different now if you're allergic to peanuts or something like that. Same thing. It's not the peanut really that killed you. It's your immune system that killed you because it got far too excited trying to protect you from peanuts. So we'll talk about that. Hypersensitivity, and we'll talk about all of that. You need to know about that. The other one you need to know about are prions. So prions, equally, they're not alive. They're not cellular. They are just proteins. So they don't even contain genetic material. They are just a protein that is misshapen. There's a fairly famous example of a prion disease, which probably predates you guys. I don't reckon you're old enough, but you will have heard of it. Do you know what it is? It happened in the UK quite some time ago, in the 90s.
The potatoes.
No, it's much more beef related. In fact, it was quite specifically beef related.
Mad cow.
Mad cow, yep. So mad cow disease is a prion disease. And in us, it's CJD, Creutzfeldt-Jakob disease. We'll just call it CJD because it's easier to say. Really nasty. Like it is literally just a protein. So it's not like antibiotics won't work, antivirals won't work. It's just a protein. And how do you target a protein in terms of treatment? The reality is we don't. If you get CJD, you will die. There's not any other potential outcome. Basically, it turns your brain into Swiss cheese progressively, punching holes in your brain. They call it spongiform encephalitis, which is because your brain looks like a sponge.
So you probably shouldn't eat meat.
No. Even if you got it now, there's no treatment for it.
No, there's no treatment for it. So it's scary. Like it's scary, but at the same time, it's incredibly rare. One of the issues that led to that was they were feeding offal, so like offcuts from sheep and stuff like that to cattle, which led to them picking up the prion, and it was called mad cow disease because it affected their brain and they effectively went mad. And then when we ate the beef, it was doing the same thing to humans as well. But even now, I don't think it's changed. But if you're in the UK during, I can't remember the years, but a certain period of time, if you're in the UK, you're not allowed to donate blood because we don't know how long it might hide in you before it's there, and we don't know that it can be transmitted through blood transfusion. Like we're not sure that it is, but we're not sure that it isn't. So we don't want to accidentally sort of transfuse a whole bunch of people and give them CJD or mad cow disease. So yeah, we're very careful with it.
So we should all be vegetarian.
Sure.
Can you test for it in your blood and like see if you have it?
They can do tests for it. Yep. So they'll do tests for it, both brain scans, and there is a blood test for it. I don't know how accurate it is though. Look how.
Can you test your meat? Will you eat it?
Not easily, I don't think. I don't know, is the honest answer. I'm not sure. But certainly there's no evidence of it having been in Australia, and there's no reason that we like there's a lot of it's illegal to feed offal to animals for that reason. So there's no reason to expect that we would expect there to be an outbreak or anything like that. But you want to be careful. So viruses basically are genetic material wrapped in a protein envelope, and prions are just a protein, not even genetic material. So that's the biggest thing. And they're non-cellular. Those are the two that you need to know. Okay, that's pretty much the end of that chapter. So we're going to do some questions together. Do you guys have your textbooks with you?
Yeah.
Excellent. All right, so let's start here though. You don't need the textbooks for this little bit because these questions are pretty straightforward. So question 1, tell me the answer, A or B?
A.
Yep, happy with A?
Yeah.
Yep, excellent. All right, see if we can fill in this sentence together. So just have a read through it and we'll work through it together. So how do we feel about filling in those blanks? So let's start with the first word. What's the first word?
Antigens.
Yeah. So antigens are molecules that are recognized by the immune system as either foreign or self. In humans, self-antigens take the form of?
MHC proteins.
Yep, MHC proteins. All right, so our self-antigens, remember, are MHC markers or MHC proteins, which show a high degree of variability between individuals. Okay, so if
non-self
antigens are detected, the immune system's activated and launches an attack response. If the immune system mistakenly attacks self cells, this is known as
autoimmune disease.
Autoimmune. Beautiful. That's purely just about your ability to use the key terminology in context. That's what that question is really trying to test out and suss out. Excuse me, because there's a lot of new words, as there always is in biology. Using them in context is obviously going to be important. So, yep, that's good. All right. So I'll alternate between the two of you. So Flick, it's just because you're on my left on my screen. I don't know if it's the same for you guys, but you're on my left on my screen. So I'll get you to go first. Cellular or non-cellular for protozoa?
Cellular.
Yep. And Paige, bacteria?
Non-cellular.
Bacteria or non-cellular?
Aren't bacteria not in your body? Oh no, they are cellular.
They're cellular, yeah. And you've got more bacterial cells in your body and on your body than you have cells that make you. You're absolutely full of bacteria, swarming with them, in fact. Hopefully, because otherwise you'd be really, really sick. That forms part of your first line of defenses, which we're actually going to look at in the next chapter. Okay, so Flick, viruses?
Non-cellular.
Non-cellular. Paige, worms?
I don't know.
Cellular.
Yep, cellular, yeah. There's only 2 non-cellular, so that's probably the easiest way to remember. So prions, Flick?
Non-cellular.
Yeah, non-cellular. Sorry, just trying to work out some stuff. And either one of you can answer the last one. Fungi?
Cellular.
Cellular, yeah. So the only 2 that are non-cellular are viruses and prions. Basically, if you remember that everything else is cellular, you're going to be fine. Right, this is a little bit more detail. You won't need to know a huge amount of detail, but this is now taking a description of our bacteria, of our pathogen, I should say, and just a really brief description of what the pathogen is like. So we'll work our way through from top to bottom. So 1, 2, 3, 4, or 5 down here. Bacterium, which one would you say is the best description of bacterium? 1, 2, 3, 4, or 5? And I know we didn't spend a lot of time on this, so that's I've sort of skipped through this. 2. Are they eukaryotic or prokaryotic?
Prokaryotic. So 4.
Yeah, it's 4. Yeah, so 4 is the best description for that. So protozoa.
Would that be 2?
Yeah, so protozoa are eukaryotic. They're more complex organisms, and they obviously can cause disease. Fungus.
3.
Yep, 3. Long branching filaments called hyphae is a typical characteristic of fungi. Prion.
5.
Yep. So prion is abnormally folded proteins. Yep. And last one, our virus.
1.
1. Yeah. Those 2 are probably good for you to remember in a bit more detail. The virus and the prion, particularly viruses in the fact that they are intracellular pathogens, that they literally, a virus outside a cell can't do anything, right? It literally is not going to cause you any problems. It's only when it gets into a cell that it causes problems. That presents a unique challenge too for the immune system, because your immune system, remember, it's going to recognize self. If you've got a virus living inside one of your cells, it's hidden. The virus is hiding from the immune system inside one of your cells. I've already touched on the fact that you can get changes to the MHC marker, and that sort of helps the immune system along. But equally, largely, your body is going to kill part of itself to try and kill the virus. And so it presents some unique challenges. And so it's something that we spend a bit of time having to unpack and talk about. So understanding that it's non-cellular, understanding that it lives inside cells, that's important. But we'll talk about that more. If you've got your textbook, I didn't bother copying and pasting this one in because there's quite a bit of information there. So if you have a look at page 361, and then we'll just work through questions 6 to 11 together. So I'll give you a couple of minutes to read that. It's about hepatitis A. Okay, so we've read through it.
Yeah.
Yep, cool. All right, so question 6. Infected individuals consumed oysters from Wallace Lake, all A or B?
B.
It is B, yes. It's unfortunate they ate poo, but there you go. That's what happens.
I have a question.
Yes.
I got gastro once from when everyone was putting their poo in the river.
Right, yes.
Does that mean I could have got hepatitis?
You could have. There's a lot of other things you could have got as well. But yeah, you could have. It's passed on by fecal oral route, so that absolutely could have happened. If it obviously requires someone to have hepatitis A to pass that on. You probably know because you'd end up, well, potentially jaundiced. So we talked before about babies being jaundiced with that yellow coloration to the skin. If your liver isn't functioning properly, that happens to you as well, because the liver is responsible for removing bilirubin. If it's not working, you tend to get this build up and you get this yellow tint to your skin and potentially your eyes as well. Depends how severe it is though. But yeah, it could have. But I mean, gastro, it could have been Giardia, it could have been like, there's a whole bunch of stuff it could have been. Best not just put poo in the river for starters. But anyway, that's lovely of whoever was doing that. So,
Everyone.
Nice. Yeah, I will not go swimming in the river ever. So we'll keep
Are you
going.
serious?
What?
So you never go swimming in the river?
Well, I haven't, no.
What?
I grew up in Melbourne, so I went swimming at the beach.
You're so weird.
Yeah, no, it's weird that I didn't swim. Where I grew up in Frankston, you didn't go to Frankston. You're meant to go to Cranbourne anyway, not Frankston. But Cannock Creek, which would be the closest thing they have to a river there, you wouldn't go swimming in that. You'll get hepatitis B because you'll stab yourself on syringes and stuff. No, you go swimming at the beach. So yeah, no, I haven't gone swimming.
You have to go swimming at the river.
Sure, but
That
there's no
would be
reason.
funny watching you try and kneeboard.
That probably would be hysterical. Yes, it's possibly something you're never going to see. Sorry about that. But anyway, all right. So getting into the immunity of it. So surface proteins of hepatitis A can be detected by the immune system as
A.
A, non-self. Great. All right. This is where we get into the nitty-gritty. And these are the styles of questions you might encounter. So hepatitis A is considered to be which one of those options? And do it by process of elimination. But what sort of pathogen is it?
It's non-living, isn't it?
Yeah, it's a virus, so it's not
It has
living.
to be A or C.
Yeah, A.
Yeah, it's A because it's non-living. You're right, but it's also, remember, viruses are non-cellular, so A is the correct response. Good. This then drills into that question we looked at before. It's sort of just rewording it in a different way. The structure of hepatitis A is comprised of.
B.
Yep. What do you think, Paige?
I got no idea.
All right, well, let's work through the options. Let's talk about what each one is. So a tough cell wall with a flagella for locomotion. Keeping in mind this is a virus. Does that sound
No.
right? No. So that would be like a protozoa that they're talking about there. Genetic material enclosed within a capsid.
What's a capsid?
It's the protein coat.
Around the.
It's the protein coat around the genetic material. Yeah, which is what a virus is. So that's B. C, the branching filament's called hyphae. That's fungi, and abnormally folded proteins is prions. So that's that one. I need to bring the graph back up because I can't look at it. This is purely just about your ability to read and interpret and understand data. That's pretty much all these questions are. So based on the graph, in January of 96, there were how many hospitalizations? I need to have a look myself.
Like 50.
Yeah.
Ish.
0 to. Oh wait.
Oh, what?
Yeah, so you're looking at the options. You've got 3 to 400, 1 to 200, 2 to 300 notifications. So you've got a few different
So
options.
D.
D 0 to 100 notifications in January of 96.
Oh.
That'd be C.
Oh, I thought we had to do hospitalizations. Sorry.
No, the options, yeah, it's kind of sneaky the way they've done that.
Yeah.
A little bit cheeky.
It is. It's a little bit cheeky. Yeah,
In
so
C?
it would have to be C, wouldn't it? But even that, I'm not convinced. It would have to be C. That's the closest. The hospitalizations are below 100 the whole way through in January of 96. So, so A isn't right because it's not 3 to 400. It's less than 100. And again, B is not right because it's less than 100. So it can't be A or B. There's clearly more than 0 to 100 notifications. So by process of elimination, I'd be saying C. But even that, is it 2 to 300? I would have said it's under 200. That would be my answer. I'd be saying it's under 200. But anyway, that's not an option. All right, so let's. I would say C. If you said C, you're going to get the answer right. If I put that on a sack, I will not put a question like that on a sack. So let's not get too hung up on it. And based on the graph, if notifications associated with the Wallace Lake outbreak were excluded, there would be how many notifications? So if we excluded the notifications from the outbreak.
And what?
That doesn't make sense.
All right, so the outbreak occurred in 1997. And there was a big spike, so that you can see that big spike where you've got nearly 700 notifications of hepatitis.
Mm-hmm.
What they're asking you is if you look at 97 and you got rid of that spike, approximately how many would there be?
It would be A.
Yeah, it would. I'd say to be A. Yeah, you'd expect there'd be a flat line between 1 to 200, and I'd be answering A for that one as well. Yeah. Not those graph questions. It's a bit hard to read that graph. This one, I'm not going to get you to do now. This one I am going to ask you to do as a bit of homework. I need to bring you guys back up so I can see you. There you are. Because I had the textbook open on my screen and I couldn't see you guys. These questions are, I'm hoping, going to be very useful. So I'm going to set some of these, not all the time, but I'll set some of these as homework and I'll post them on here. You can actually respond to them on a Word doc or however you want to do it, but I'll post them on here. But if you look on here, some of this stuff is just review of this chapter, right? So define what we mean by disease. MHC complexes are types of protein embedded in cell membranes. Explain how a single gene can produce many different proteins. That's not specifically about immunity anymore, is it? Can you guys answer that question at the moment? How can one gene lead to many different proteins? That silence concerns me greatly. I want you to think about stuff you've already done, stuff that you already know, stuff that you actually answered questions on in a SAC and everything. So I'm going to annoy you and say, I know that you know this. That is, honestly, the best eye roll that I've seen there, Paige. Well done.
From it, like, evolving.
Now, think about how can one single gene lead to more than one protein product? What's the name of the process?
What does it start with?
No, I'm not going to tell you that, but I'll tell you instead. I'll mention two phrases, exons and introns, and then I want to see if that triggers it off in your mind.
Splicing.
Yep. Give me another word in front of splicing.
Alternative splicing.
Yeah, so it's alternative splicing. That's what that question's asking there. I know that you both know it. We just need to trigger it in just the right way. So these questions here are going to probe your memory about some of the other stuff related to, like, these are the sorts of questions you can expect to encounter on the exam, right? But the first thing there you need to do, don't get distracted by that first line. MHC complexes are a protein embedded in cell membranes. Yeah, great. That's an introductory sentence. Explain how a single gene can produce many different proteins. Your first step there is, what is the question asking you? Is it actually asking you what an MHC protein is? No, don't care. It's just asking you, it's a protein, full stop, that's it. Then it's asking you about really RNA processing. That's what it's asking you about,
So
of which alternative splicing is part. Sorry?
The answer to that is alternative splicing.
Yeah. Yep.
Is that all we have to say?
You need to explain how a single gene can produce many different proteins. So you'd say that one gene can contain exons and introns, and then as a result of the process of alternative splicing, exons can be put together in various combinations leading to various protein products. Yeah, so just dot points is all you need, but it's only a 1 mark question. So I would actually argue that question in the exam would be worth more than 1 mark because there's a few steps to that. I haven't actually looked at what they've said in the back of the textbook, but that, to me, you need to say a few things. Explaining means naming the process, then all you need to do is name it. But when they say explain, you actually need to, saying alternative splicing doesn't explain it. Due to the risk of infection, individuals taking immunosuppressants often take antibiotics to prevent the occurrence of bacterial infections. Again, we won't go through this now, but this is what I'm going to leave with you guys, and this is the reason why I'm doing it. Rifampicin is a type of antibiotic which inhibits RNA polymerase. Right, so hopefully that's starting to trigger something. What I want you to tell me at the moment, not the answer, I just want you to tell me, read C part 1 What's the question actually asking you to do?
Identify and outline the process in which that inhibits.
Yeah, so what process? What's it actually asking you to explain? Identify and outline the process. So let's start by identifying it, because that's the first thing it's aiming to get you to do. We're not going to go through, I don't want to go through the outline now. I'm going to leave that with you guys as homework. But what's the name of the process?
RNA polymerase.
No, that's the name of the enzyme that drives the process. What does RNA polymerase do?
Reads it.
Reads what?
The DNA.
Yep, it does read the DNA. It's through the process called?
Transcription.
Yeah, transcription. That's what it's asking you. So the name of the process is transcription. These questions are going to be useful because, yes, it's going to start tying in immunity, but it's going to call back to things that you need to know from Unit 3, which you are going to need to know.
I can't remember anything from Unit 3.
Yeah, well, you told me that when you had to go at the practice exam, which we need to work through with all of you, really, because I've handed out the practice exam and then we've done nothing with it. So we need to do that. But these questions will help with that, right? Because, again, you know it, like that information is rattling around inside your head. But the first step is, for you guys, what's the question actually asking you to do? Is it asking you what the antibiotic does? No, it's told you what the antibiotic does. I don't care about that. It's telling you that it inhibits RNA polymerase, which then it's quite a good question because it's then going to probe your understanding of how RNA polymerase, what does it actually do? Do you remember what that enzyme is? Do you remember the process that it's involved with? Do you remember the steps involved in transcription? So it is quite a good question. So I've got it in a Word document. I'm going to post that on here and ask you guys to have a look at that. That should be it. That's worked out well at the end of the first period.
Now let's see a dog.
Maybe in a minute. Excuse me. So hopefully we feel okay. Like if we were to rate ourselves on the success criteria here, feeling okay with what we mean by an MHC receptor. Yeah. Flick's nodding, you're doing nothing, Paige, so I don't know. Well, tell me, what are the two types of MHC receptor?
Number 1 and number 2.
Yep, where do I find number 1?
In all what's the word?
Yeah, all nucleated cells. Yep.
Yeah.
Yep, so every nucleated cell has MHC1. Where do I find MHC2?
In the immune.
Yeah, in immune cells is the only place I find it. Right. And Paige, this one's specifically for you, just because you've said that you had no idea a couple of times when we asked this. So I'm hoping that will change now. Name the 2 non-cellular pathogens for me. So just the 2 non-cellular.
Viruses.
Yep.
And the P1.
And the P1, yeah. So prions. Yep, I'll pay that. I'm happy with the P1. It'd be better if we write the whole word if you have to on the exam, but I'm happy with that. That's fine. Great. All right, cool.